Welcome back to my channel Adhyan with Flow. In previous video, we discussed about transcription and types of RNA. Ribose nucleic acid. How many types? 3. rRNA. We call it as a ribosomal RNA. Are you familiar with this term, ribosome? Next is mRNA. We make mRNA through transcription, DNA to RNA, remember? Messenger RNA. Next is tRNA. This is the transfer RNA. This is the structure of tRNA. Some students ask, like, ma'am, DNA and RNA, same phosphate, yeah. Sugar group, yeah. Purine and pyrimidine. So why DNA is genetic material but RNA is not genetic material? Just because they are double helical and this is a single helical doesn't mean that they are genetic material and they are not. Guys, remember, they contain A, G and C and T. But they contain what kind of nitrogen base? A, G, C, but uracil. Why, guys? Why they contain uracil? Many students ask, why, ma'am, uracil is present instead of thiamine? Guys, DNA, where they located? This is a cell, here is nucleus and they are present in the nucleus. But the function of RNA is what? DNA produce RNA like mRNA. Now what is the function of mRNA? They code for protein. Yeah, they also contain message. Same, DNA also contain message and they transfer generation to generation for a long time. But they contain message. Yes, this is true that they also contain message and they transfer their message, but for a little bit time, only 10 minutes. This is the lifespan of RNA, guys. So that's why DNA is genetic material and RNA stability also less there. Because of uracil, their stability is maintained because they come out of the nucleus and they have to done many functions. DNA, what they do? They transfer genetic material to generation. They also transfer message, but only for formation of protein, particular enzymes. Not they pass their cell generation to generation because their lifespan is already little bit. Okay, you have to clear this thing. RNA, they contribute formation of ribosome. mRNA, they contain message that what kind of protein they want to form, what kind of enzyme, what kind of hormone, what kind of steroids, whatever they want to do. tRNA helpful in translation. They are supporting element to formation of protein. Without tRNA, without rRNA, without mRNA, protein never form. Yaar. You can't make protein without ribosome, without mRNA, without tRNA. You can't get protein. So DNA to RNA formation, we call it as a transcription. Further, mRNA, what they do? Protein. So this process is transcription, this process is translation. So we are going to study about translation, means how mRNA form protein. So for translation, you have to remember RNA. What? Oh, wait. Their types and their types of functions, like what rRNA do, what mRNA do, what tRNA will do. Without these three types, protein will not form. Okay, let's start with translation. In this phenomena, we get RNA to protein. If I say transcription, then what thing you explain? How RNA will form through the DNA. Now, if I ask, tell me something about translation, ma'am, protein will get from the mRNA. Let's start. First step is what? Charging of tRNA. You can say amino acylation of tRNA. This is simple, guys. What you mean? What you understand by charging of tRNA? I am talking about this. tRNA. This is the tRNA. If I add amino acid on tRNA, it means tRNA is charged. Now he is ready for translation. I repeat, if someone asked what is charging of tRNA, what is amino acylation of tRNA, what do you reply them? That simple, when tRNA charged with amino acid, this kind of phenomena we can call it as a charging of tRNA. And guys, how translation will occur? this is what I am drawing here. Wait. This is what large subunit of ribosome. This is the small subunit of ribosome. Okay, who give rise ribosome? Remember, RNA. So this is the large subunit of ribosome and this is the small subunit of ribosome. In previous classes you already know that eukaryote contain ATS RNA, okay, and prokaryotic contain what kind of ribosomal RNA? 70S types. ATS divided break into large subunit unit is 60S, small subunit is 40S. We are talking about eukaryotic, and 70, 50 and 30. So this is the 60S large subunit and this is the 40S large subunit. They again further divided into many sites. They further divided into many sites like 20S RNA, 5.8S RNA, 5S RNA, 16S RNA, 18S RNA. Okay, this is not in your syllabus. Okay, relax guys. Okay. Now this is the E site, this is the P site, this is the A site. This is not in your book. Okay. Now listen to me carefully. Here tRNA will goes out, exit. Here next new tRNA will come and here polypeptide bond will form. And if you remember, what is peptide bond? Peptide bond present between two amino acids. Two amino acid bind with peptide bond. In biochemistry playlist I explain these things. Okay, you can watch. Okay, if you want detailed idea about what is amino acid, what is peptide bonds, you can switch to my biochemistry playlist and tools and techniques playlist. Next thing is what? Here peptide bond will form, here amino acid chain will form, and here one is coming here, transfer here and exit here. Again new is come here. So they make amino acid polypeptide chain and further in future they make protein. How they do this phenomena, let's discuss properly. Now first thing is what? This is the large subunit, clear or not? I am not writing here, okay, now I am not writing here. Large subunit of ribosome, this is the small subunit of ribosome, clear? Yes, very clear. Look at here, this is the E site, this is the P site and this is the A site. Look at here, here tRNA will come, here tRNA will go and here look at here, peptide bond will form. Clear or not? Let me remove. I think clear. Next thing is what? Now look at here, here is mRNA present between two ribosomal unit. Like this is the large subunit and this is the small, and here mRNA will lie down. We make na DNA to RNA, yaar. DNA to RNA. During transcription, we get na mRNA. So we want protein na after RNA. They are contributing in protein formation. How they contribute? They present it. Now they come out from nucleus. And they fit between ribosomes. They are fit. Now what happen? tRNA will come with their amino acid. tRNA will come with their amino acid. They pair with codon and codon is present in 3 set. Clear or not? Again I am removing so I can visualize you betterly. I am drawing here a tRNA with their amino acids. At this side, first tRNA will come, bind with nucleotides, have amino acid. Now this sequence mRNA will go little bit forward. Then again, next step, what happen? Look at here, this size is big, look at here and look at here, this size. They move forward, mRNA, and now you found something in this thing, and next tRNA will come. Yes or not? They bind here na, anything they A, U, G, C, T, no, not C or not T. Here is amino acid methionine, always methionine will come, and you said, why ma'am methionine is come first? Why ma'am? Because methionine have starting codon AUG. Methionine start what? Translation. So translation process, make sure that methionine come first, then they start translation process. And why methionine is responsible for starting the process? Yeah, we know that they have starting codon. AUG, they have starting codon. But except starting codon, guys, they increase little bit lifespan of mRNA, little bit lifespan of protein. Like if the lifespan of mRNA is 10 minutes, assume 10 minutes, and protein lifespan is what, 15 minutes. If our body start with methionine translation, then it increase 25 minutes, 30 minutes. Okay, so methionine, yeah, we know methionine is starting codon. They start translation, but why methionine? Because they increase lifespan of protein. Okay, now assume this protein is what, this amino acid what, serine. Now what happened between them? Now there is this amino acid 1 and this amino acid 2. They make peptide bond. They make peptide bond. Now what thing is next? Now what thing is made? Now this move thisward, this move thisward with no amino acid, and this tRNA get methionine first amino acid, then second is serine, then another tRNA come with their, assume this is the valine amino acid, and they reach here. Now again what phenomena will occur further? Now they release and they come here. This is release with no amino acid, and they look at here. Now this reach to this position, and they make bond with, when new tRNA will come now, look at here, they make methionine, serine, valine, and here new tRNA will come. New tRNA will come and sit here with another, assume this is the leucine. So further chain will come, chain will go, chain will make, chain will merge. Till then, when stop codon is not coming. Till then, when stop codon will not coming. After stop codon will come and sit here now, all polypeptide chain will release from ribosome, and here we get a polypeptide chain, and future they form protein. So now you are understand. So this is all about translation, guys. Thank you so much guys for listening me. Thank you so much.